everyone, welcome to Neuroscience for Young Minds. In this presentation, I'll be explaining the structure of the brain and how it is intertwined and what is new in neurodegenerative sciences and a lot of more fun facts. So let's start. So how do we start? From where do we start? So when we do not know from where do we start, let's start for something which is very small. And what can be smaller than a cell in our body? So let's start from the cells. So in human body, we have multiple types of different cells. For example, we have blood cells, heart cells, muscle cells, pancreatic cells, kidney cells, liver cells, bone cells, lung cells, and last but not the least, brain cells. And these are not all, there are many more types of cells. What this picture is trying to tell you that we have different organs in our body which are composed of different types of cells. As you can see pictorially also, the cells do not look alike. And when they do not look alike, do you really think they work alike? No, they don't. So that means that a specific organ has a specific job to do, and they do that specific job because of how they look and how they are made up of. So in this PPT, we will be focusing on specifically brain cells because it is all about neuroscience. So now let's learn about what is a brain. So the brain is the most important organ you have. But who said that? According to the brain, right? But joking apart, brain is actually the most important organ we have in our body. So let's dive deep into the subject and learn more about the brain. So these brain cells are called as neurons, and they're also called as nerve cell. So a neuron is a nerve cell, and the brain is made up of approximately 86 billion neurons. Equal number of neuronal and non-neuronal cells make the human brain an isometrically scaled up primate brain. So neurons are similar to other cells in body in some ways, such as neuron is also surrounded by a membrane like other cells in our body. Neurons have a nucleus that contains genes. Neuron contains cytoplasm, mitochondria, and other organelles like other cells in our body have. So these are some of the reasons why it is common to the other cells which are present in our body. So now let's look at this, how neurons look. So neuron looks like this, which is on the right side of your screen. Do you see this star-shaped structure? Yes, this is star-shaped structure, and the tail afterwards is in totality called as a dendrite. And this is the complete whole structure. Starts with a star sign and then ends with a tail with tentacles. This tail is called as axon, and this axon eventually goes and binds to another star-like shape, and whenever these tentacles they bind to another star-like shapes, this contact is called as a synapse. This is called as soma, and this is called as an axon, and this protective layer over the axon is called as myelin sheaths. So, although I just told you that neurons are similar to other body cells, but they are definitely different from other cells in the body, and some of the examples are neurons have specialized projections called dendrites, which I told you, the ones which look as stars, and axons. So the star shape is called as a dendrite, and the tail part is called as an axon. An axon is in turn protected by a protective sheath called as myelin sheath. So dendrites bring information to the body cell, and axon take information away from the body cell. So receiver of this kite-like structure is the dendrite, and the one which passes on the information to the next dendrite is an axon. So dendrite, they bring information. Axons, they send information to the axons which are underneath them. These neurons communicate with each other through an electrochemical process, and the neurons form specialized connections called synapses and produce special chemicals called neurotransmitters that are released at the synapse. So, I was telling you about this synapse. So, in this synapse, they come into proximity, touch each other, and then the neurotransmitter is transferred from one axon to the from the axon to the other dendrite, and this is how the synapse happen, and this is how the current in the neurons is floated. Yeah, so you guessed it right. Whenever the neurotransmitters work and whenever the signal is passed on, it looks like this, which you can look at right now. They look like the sparks of electricity. So now let's learn more about neurons or nerve cells. So neurons come in many different shapes and sizes. Some of the smallest neurons have cell bodies that are only 4 microns wide, and some of the biggest neurons have cell bodies that are 100 microns wide. And remember that 1 micron is equal to 1/1000th of a millimeter. Alright, so a human brain has approximately 86 billion neurons, and neurons are specialized to carry messages through an electrochemical process, and that process is called as action potential. So let's understand again. Neurons are surrounded by a cell membrane. Neurons have a nucleus that contains genes. Neurons contain cytoplasm, mitochondria, and other organelles. And neurons carry out basic cellular processes such as protein synthesis and energy production. Now, let's learn about the action potential. So, much of what we know about how neurons work in our body, it comes from the experiment on the giant axon of the squid. So, can you see this sea creature? The sea creature is called as a squid, all right? And this squid has giant axon. I just showed you an axon, right? Axon was the tail part of our neuron. And this particular creature, since it is so long, so it has a very giant axon which extends from the head to the tail of the squid and is used to move the squid's tail. Imagine how long it is. You can see the person in next to this giant squid, right? So how giant is this axon? Let us understand. So it can be up to 1 millimeter in diameter, which is very easy to see with the naked eye. But can we see it in case of humans? Not a chance. So that is why much of the work we know about how the neurons work, they come from the experiments when we do experiments on sea creatures like them. So now these neurons, they send messages electrochemically, and this means that chemicals cause an electrical signal. So chemicals in the body are electrically charged, and when they have an electrical charge, they are called as ions. So the important ions in the nervous system are sodium and potassium, and both of them has one positive charge, and calcium, which has two positive charges, and a chloride, which has one negative charge. And there are also some negatively charged protein molecules also. So these are the important ions which are present in the nervous system. So it is also important to remember that nerve cells are surrounded by a membrane that allows some ion to pass through and blocks the passage of the other ion. So this type of membrane, which is very selective, is called as a semi-permeable membrane, which only allows certain ions to pass and restrict others. Now let's understand what is the resting membrane potential. So whenever a neuron is not sending any signal, we call it at rest. And when a neuron is at rest, the inside of the neuron is negative relative to the outside part of the neuron. So although the concentrations of the different ions attempt to balance, on both the sides of the membrane, they cannot, because the cell membrane allows only some ions to pass through the channels. So when the neuron is at rest, potassium ions can cross the membrane very easily. And while the neuron is at rest, chloride ions and sodium ions, they have more difficult time to cross the semi-permeable membrane. So the negatively charged protein molecules in this picture, let's say those are A negative. So the negatively charged protein molecules, A negative, inside the neurons, they cannot cross the membrane. So in addition to these selective ion channels, there is a pump that uses energy to move 3 sodium ions out of the neuron every 2 potassium ions it puts in. So whenever 2 potassium ions come in, 3 sodium ions have to go out, and this is how they keep the balance in the neuron across the neuronal cell membranes. So finally, when all these forces balance out and the difference in the voltage between the inside and the outside of the neuron is measured, that particular voltage is called as resting potential. So the resting membrane potential of a neurons is around negative 70 millivolt. This means that inside the neuron, it has 70 millivolt less than the outside. And at rest, there are relatively more sodium ions outside the neurons and more potassium ions inside the neurons. So this is how the ion concentration gradients are whenever and across the cell membrane whenever the neuron is at a resting state. That is why it is called as resting membrane potential. So now you can see whenever you will measure it, it will come out to be negative 70 millivolt. And yes, this is how we measure it. So, these action potentials are caused when different ions cross the neuron membranes. So, a stimulus first causes the sodium channels to open. So, first of all, sodium channels open. And because there are many more sodium ions on the outside and the inside of the neuron is negative, because I just told you that when the neuron is at resting state, the inside of the neuron is negative compared to outside part of the neuron. All right. So, because there are so many sodium ions on the outside and the inside of the neuron is negative relative to the outside, sodium ions, they try to rush into the neuron. So, remember, sodium has a positive charge. So, the neuron becomes more positive and becomes depolarized. So, now it takes longer for the potassium channels to open. So, when they do open, potassium rushes out of the cell, reversing the depolarization. So, this act of polarizing and depolarizing, this keeps the triggers and stimulus on. So also at about this time, sodium channels start to close. So this causes the action potential to go back towards negative 70 millivolt, which is a repolarization. So the action potential actually goes past negative 70 millivolt because the potassium channels stay open a bit too long. So gradually the ion concentration go back to resting levels and the cell returns to negative 70 millivolt. So what it is trying to tell you that while it is in process, while the ions are moving in and out of the cell and are in the process of depolarizing and repolarizing, sometimes when you will check the voltage in between, it may be more or less than negative 70 millivolt. But when the process is closed and this neuronal cell is finally at peace, it will always come to be as negative 70 millivolt. Yes, you got it right. So that means our neuronal cells, our brain is actually always charged. Try and touch it sometimes. Alright, so now let's learn about classification of neurons. So neuron can also be classified by the direction that they send information. So for example, there are sensory, motor and interneurons. So a sensory neuron is also called as efferent neurons and they send information from sensory receptors. For example, skin, eyes, nose, tongue, ear, and they send it toward the central nervous system. Right? And then we have motor neurons, also called as efferent neurons, and they send information away from the central nervous system to muscles or gland. And then we have interneurons, and they send information between sensory neurons and motor neurons. And most, interneurons, they are located in the central nervous system. Now let's talk about neurons and neuroglial cells. So the one in yellow color are the neurons, which we just talked about. So you can see it has a dendrite and the axon part. And the other nucleoglial cells, which you can see apart from neurons in this picture, are microglia, oligodendrocytes, and astrocytes. So let's see what microglia is. Microglia you can see here in the brown color. So these microglia, they are located throughout the brain and spinal cord and they act as the first and main form of active immune defense cells in central nervous system. So they protect us against any type of attack. Now comes oligodendrocytes, which are in blue color. So they provide support and insulation to axons in the central nervous system and these are also equivalent to function performed by Schwann cells in peripheral nervous system. Now, astrocytes, which can be seen in green color. So these type of neuroglial cells, they provide trophic support to neurons in central nervous system and have functions such as neurotransmission, metabolite and electrolyte homeostasis, cell signaling, inflammation, and synapse modulation. Now let's learn a little bit about neuroanatomy. So, the central nervous system is divided into two parts, the brain and the spinal cord. The average adult human brain weighs 1.3 to 1.4 kg approximately, and the brain contains about 86 billion nerve cells, which are neurons, and trillions of support cells called as glia, which we just discussed. The spinal cord is about 43 centimeter long in adult women and 45 centimeter long in adult men and weighs about 35 to 40 grams. The vertebral column, the collection of bones, which is called as a backbone that houses the spinal cord, is about 70 centimeter long. Therefore, the spinal cord is much shorter than the vertebral column. Now look at this. This is a Stegosaurus. So a Stegosaurus dinosaur weighed approximately 1600 kg but had a brain that weighed only approximately 70 grams. Therefore, the brain was only 0.004% of its total body weight. And in contrast, an adult human weighs approximately 70 kg and has a brain that weighs approximately 1.4 kg. Therefore, the human brain is about 2% of the total body weight. This makes the brain to body ratio of the human 500 times greater than of the Stegosaurus. Isn't it wonderful? So you can now proudly say that my brain is bigger than your brain, Stegosaurus. Alright. So what does the peripheral nervous system do? It connects the central nervous system to the organs, limbs and skin, and it carries sensory and motor information to and from the central nervous system. It allows brain and spinal cord to receive and send information to the other areas of the body, and it also regulates involuntary body functions like heartbeat and breathing. So now this nervous system is divided into two parts, peripheral nervous system, central nervous system. Central nervous system comprises of brain and spinal cord, which is easy to understand. Peripheral nervous system is little complex. It is in turn of two types, autonomic nervous system and somatic nervous system. Now this autonomic nervous system is in turn divided into two, which is parasympathetic nervous system and sympathetic nervous system. And somatic nervous system is also divided into two, which is sensory and motor. So let's see what is somatic nervous system. So the somatic nervous system consists of peripheral nerve fibers that send sensory information to the central nervous system and motor nerve fiber that project to skeletal muscle. So the picture above shows the somatic motor system. So the cell body is located in either the brain or the spinal cord and projects directly to a skeletal muscle. So the moment you're going to touch a candle, the effector muscle is going to send the signal quickly to the brain, and that is how you develop your reflex actions. Now let's talk about the autonomic nervous system. The autonomic nervous system is divided into three parts. First is sympathetic nervous system, then the parasympathetic nervous system, and the enteric nervous system. The autonomic nervous system controls smooth muscle of viscera, which means internal organs and glands. So the picture above shows the general organization of the autonomic nervous system. So the preganglionic neurons is located in either in the brain or in the spinal cord, and this preganglionic neuron projects to an autonomic ganglion, and the postganglionic neuron then projects it to the target organ. So notice that somatic nervous system has only one neuron between the central nervous system and the target organ, while this one, the autonomic nervous system, uses two neurons. Right? So here are the examples on the right hand side of the screen of parasympathetic nerves and the sympathetic nerves and how they work. So parasympathetic nerves, they reflect in rest and digestive states, and sympathetic nerves, they reflect when there is a condition of fight, and flight. The enteric nervous system is the third division of the autonomic nervous system that you do not much hear about. The enteric nervous system, ENS, or intrinsic nervous system, is one of the main divisions of the autonomic nervous system and consists of a mesh-like system of neurons that governs the function of the gastrointestinal tract. And if you will, go to the research, journals, you will see that some new studies have also come up which say that gut is the first brain of our body. Isn't it interesting? So this is a very gutsy move for a brain. So now this is how the central nervous system and the PNS nerve system are depicted in this figure, and I hope this is pretty much clear to you that which parts are comprised of CNS and which parts are comprised of peripheral nervous system. So once again, nervous system is divided into two parts, central and peripheral. Peripheral is divided into autonomic and somatic. Autonomic is which communicates with internal organs and glands, and somatic nervous system is which communicates with sense organs and voluntary muscles. And autonomic is in turn divided into two parts, which is sympathetic and parasympathetic. And the somatic nervous system is also divided into two parts, which is sensory and motor. Sensory is efferent nervous system, and motor is afferent nervous system, wherein the brain tells the body what to do. So some differences between the PNS and CNS are: in the central nervous system, collection of neurons are called as nuclei, and in the peripheral nervous system, the collection of the neurons are called as ganglia. So in the CNS, collection of axons are called as tracts, whereas in PNS, the collection of axons are called as nerves. In peripheral nervous system, neurons can be functionally divided in three ways: sensory, cranial, and somatic. So sensory is which carry information into the CNS from the sense organ, or motor efferent type which carry the information away from the central nervous system for muscle control. And cranial is when it connects the brain with periphery or spinal and connects the spinal cord with the periphery. Somatic is when it connects the skin or muscle with the central nervous system, or visceral connects the internal organs with the central nervous system. So now let's talk about the brain structure. So this is the image of the real brain in the hands of Medico. And these are the parts of the brain, major parts of the brain. First is called as cerebrum, the other is called as cerebellum, and the last one is called as brainstem or medulla. And the picture on your right is the actual image of the human brain. So from a top view, notice how the brain is divided into two halves, right? And they are called as hemisphere. So each hemisphere communicates with the other hemisphere through the corpus callosum, which is the bundle of nerve fibers. So this corpus callosum, it keeps both the hemispheres intact. And another small fiber bundle that connects the two hemispheres is called as anterior commission. So here is this picture on the right hand side of your screen, which is hilarious, but it conveys the message clearly that corpus callosum is the great mediator between both the hemispheres of our brain. So now the brain functions and their segregations by their lobes. So there are frontal lobe, parietal lobe, occipital lobe, cerebellum, brainstem, and temporal lobe. And let's see what functions they perform. So this frontal lobe, which is depicted in orange color, it is involved in problem solving, judgment, inhibition of behavior, planning, anticipation, speaking, emotional expression, awareness of abilities, and likewise. The parietal lobe, which is depicted in yellow color, has the sense of touch, taste, smell, differentiation, size, shape, color, visual perception, academic skills, maths calculation, reading and writing. The occipital lobe, which is displayed in green color, is more utilized for recognition and reading and perceptions of the things. The cerebellum is more involved in the coordination of voluntary movement, balance, equilibrium, and some memory or reflex motor acts. The brainstem is responsible for the sense of balance, reflex to seeing and hearing, blood vessel control, breathing, heart control, digestion, heart rate, swallowing, blood pressure, temperature, etc. And the last one, which is the temporal lobe, which is depicted in sky blue color, is used for understanding language, organization and sequencing, information retrieval, musical awareness, memory, hearing, learning, feelings. So the nervous system can also be divided in these different terms. First is teleencephalon, then diencephalon, then mesencephalon, then metencephalon, and myelencephalon. So now the nervous system is divided into PNS and CNS. CNS comprises of brain and spinal cord. Now this brain is divided into forebrain, midbrain, and hindbrain. The forebrain in turn is divided into telencephalon, diencephalon, and midbrain is divided into mesencephalon and metencephalon, and hindbrain is divided into myelencephalon. And the cerebral cortex, basal ganglia, hippocampus, and amygdala. These are the part of telencephalon, thalamus, and hypothalamus. They fall in diencephalon, tectum, and tegmentum. They fall under midbrain and under mesencephalon, and pons and cerebellum. They come under metencephalon, and medulla, which comes under myelencephalon. This autonomic nervous system and somatic nervous system comes from peripheral nervous system, and the autonomic nervous system is divided into two, like we know, sympathetic nervous system and parasympathetic nervous system. Now let's learn about brain structures. So the cerebral cortex is involved in thought, voluntary movement, language, reasoning, and perception. Cerebellum is involved in movement, balance, and posture. Brainstem is used for breathing, heart rate, blood pressure. Hypothalamus is used for taking care of body temperature, emotion, hunger, thirst, and circadian rhythms. Thalamus is for sensory processing, movement, and limbic system is for emotions and memory. So the limbic system is a group of structures that include amygdala, the hippocampus, mammillary bodies, and cingulate gyrus. And these areas are important for controlling the emotional response to a given situation. And the hippocampus is also important for the memory. So, like we said, hippocampus is important for learning and memory, and basal ganglia is for movement, and the midbrain is for vision, audition, eye movement, and body movement. So now the next question is, do we really use only 10% of our brains? Because people keep on telling us that we do not use our full brain, and we do not use our full brain in full capacity. We only use 10% or some part of it. So let's see. There are approximately 1 quadrillion synapses in human brain. That is, oh my god, so many zeros after one synapses. And this is equal to about 500 million synapses per cubic millimeter. Imagine. So, do you really think we only use 10% of our brain? No. So, where did the 10% myth begin? So the 10% statement may have been started with a misquote of Albert Einstein or the misinterpretation of the work of Peripherence in the 1800s. It may have been William James who wrote in 1908, "We are making use only a small part of our possible mental and physical resources," which is published in The Energies of Men, page 12. Perhaps it was the work of Carl Lashley in 1920s and 1930s that stated it. So Lashley removed large areas of the cerebral cortex in rats and found that these animals could still relearn specific tasks. So we now know that destruction of even small areas of the human brain can have devastating effects on behavior. So that is one reason why a neurosurgeon must carefully map the brain before removing any part of the brain. So tissue during operations for epilepsy or brain tumors, they want to make sure that the essential areas of the brain are not damaged. So possibly, because of the literature which has been published in the past is also contributing to the fact. And why does this myth still continue? Because somewhere, somehow, someone started this myth, and the popular media keep on repeating this false statement. So soon everyone believes the statement regardless of the evidence, and I have not been able to track down the exact source of this myth and have never seen any scientific data to support it. So according to the believers of this myth, if we could use more of our brain, then we could perform super memory feats and have other fantastic mental abilities. Maybe we could even move objects with a single thought. Imagine. So again, I do not know any of the data that would support any of this. So what does it mean to use only 10% of our brain finally? So what data were used to come up with the number 10%? We don't know. Does this mean that you would just be fine if 90% of your brain was removed? And if the average human brain weighs 1400 grams and 90% of it was removed, so that would leave to 140 grams of the brain tissue, and that is about the size of a sheep's brain. So, will it be enough to survive and to do just fine in this life? So, it is well known that damage to a relatively small area of the brain, such as that caused by a stroke, may cause devastating disabilities. Certain neurological disorders, such as Parkinson's disease, also affect only specific areas of the brain. The damage caused by these conditions is far less than the damage to the 90% of the brain. So there's no evidence and we are still in the lack of it. So perhaps when people use the 10% brain statement, they mean that only 1 out of every 10 nerve cell is essential or used at any one time. So how would such a measurement be made? So even if neurons are not firing action potentials, they may still be receiving signals from the other neurons, isn't it? So furthermore, from an evolutionary point of view, it is unlikely that larger brains would have developed if there was not an advantage. So certainly there are several pathways that serve similar functions. For example, there are several central pathways that are used for vision. So this concept is called as redundancy and is found throughout the nervous system. So multiple pathways for the same function may be a type of safety mechanism. Should one of the pathways fail, still the functional brain imaging studies show that all part of the brain function. So even during sleep, the brain is active and the brain is still being used. It is just in a different active state. So now let's talk about use it or lose it theory. So finally, the saying lose it or use it seems to apply to the nervous system. So during development, many new synapses are formed. In fact, some synapses are eliminated later on in development. So this period of synaptic development and elimination goes on to fine-tune the wiring of the nervous system. So many studies have shown that if the input to a particular neural system is eliminated, the neuron in this system will not function properly. So this has been shown quite dramatically in the visual system. So complete loss of vision will occur if the visual information is prevented from stimulating the eyes early in the development. So it seems reasonable to suggest that if 90% of the brain was not used, then many neural pathways would degenerate. However, this does not seem to be the case. On the other hand, the brains of young children are quite adaptable. So the function of the damaged brain area in a young brain can be taken over by the remaining brain tissue. And there are incredible examples of such recovery in young children who have had large portions of their brains removed to control seizures. So such miraculous recovery after extensive brain surgery is very unusual in adults. But we have some great studies which have come from the children's and they have done it. So several people have mentioned that in a movie named Lucy, which came in year 2014, it promotes the 10% of the brain myth. You see, movies are a great source of fun. Although I have seen it, I liked it, but I really do not stand by the fact that we only use 10% of our brain. So next time you hear someone say that you only use 10% of their brain, you can set them straight and tell them, excuse me, we use 100% of our brain even when we are sleeping. Yeah, that's true. So now let's talk about blood-brain barrier because it doesn't let anything slip easily. So a blood-brain barrier is a specialized interface between circulating blood and the central nervous system. So it helps protects the central nervous system by allowing only certain type of molecules to pass, while others simply bounce off. So, this blood brain barrier is actually working as a safety mechanism or a defense mechanism for our own brain. You cannot imagine how much safety is provided to our brain by our own body. And this is one of the examples. So the blood-brain barrier, it passes breathing gases, glucose, amino acids, and mood changers, and neurotransmitters here and there. And these blood-brain barrier, this membrane, it blocks highly charged materials, heavy molecules, and non-lipid soluble materials. All right, and that is the reason why, although we take so much of drugs in our system in day-to-day life, but our brain is still safe and doesn't get poisoned or toxic by the irrational use of our drugs. And sometimes, you ingest too much of alcohol also, and sometimes people are involved in some other activities also, which is not good for the brain, but brain has their own defense mechanism. For example, BBB, blood brain barrier. So this is how we are always safe. Let's learn about neurotransmitters. So, what are neurotransmitters? Neurotransmitter is a chemical substance which is released at the end of the nerve fiber by the arrival of a nerve impulse and by diffusing across the synapse or junction, and affects the transfer of the impulse to another nerve fiber or a muscle fiber or some other structure. So, these are the structures of the neurotransmitters we have. We have adrenaline, which is released in case of fight or flight, and it is also called as a fight or flight neurotransmitters. It is produced in stressful or exciting situations, increases heart rate and blood flow, leading to a physical boost and heightened awareness. Another is noradrenaline, which is a concentration neurotransmitter. It affects attention and responding actions in the brain and involved in fight or flight response. It contracts blood vessels and increasing blood flow. Next is dopamine, which is called as pleasure neurotransmitter. It gives the feelings of pleasure and also addiction, movement, and motivation, and people repeat behaviors that lead to dopamine release. Next is serotonin, which is also called as a mood neurotransmitter. It contributes to well-being and happiness, helps sleep cycle and digestive system regulation affected by exercise and light exposure. Next is GABA, which is called as calming neurotransmitter, and it calms firing nerves in central nervous system. High levels improve focus, and low levels of this neurotransmitter can cause anxiety. And it also contributes to motor control and vision. Another is acetylcholine, which is called as learning neurotransmitter, and it is involved in thought, learning, and memory, and it activates muscle action in the body and also associated with attention and awakening. Glutamate is also called as memory neurotransmitter, and it is most common brain neurotransmitter. It is involved in learning and memory, regulates development and creation of nerve contacts. Last is endorphins, which is called as euphoria neurotransmitters. It is released during exercise, excitement, and sex, producing well-being, and euphoria, reducing pain. Biologically active section is shown. So neurotransmitters can be classified as either excitatory or inhibitory. So excitatory neurotransmitters function to activate receptor on the postsynaptic membrane and enhance the effects of the action potential. While the inhibitory neurotransmitters, they function to prevent an action potential. So they are like different. They work differently. So as you can see, how the neurotransmitter is released from the presynaptic neuron and goes to the postsynaptic neuron. So this is how it is done. Now let's talk about the diseases of the nervous system. So there are more than 600 neurologic diseases, and major types include diseases caused by faulty genes such as Huntington's disease and muscular dystrophy, or otherwise problems with the way the new nervous system develops, such as spina bifida, or degenerative diseases where the nerve cells are damaged or die, such as Parkinson's and Alzheimer's disease, and diseases of the blood vessels that supply the brain, such as stroke, and injuries to the spinal cord and brain, and seizure disorders such as epilepsy, cancers such as brain tumors, and infections such as meningitis. So now let's have a look at Alzheimer's disease. So, dementia is an umbrella term that describes a collection of symptoms that are caused by disorders affecting the brain. So, it is not one specific disease. Dementia affects thinking, behavior, and ability to perform everyday tasks. And brain function is affected enough to interfere with the person's normal social or working life. And it is the most common type of dementia. Alzheimer's is the most common type of dementia, and it accounts for approximately 40 to 70% of all the dementia, and that is why we are covering it here in this presentation. So there are other types of dementias also, such as vascular dementia, Lewy body dementia, frontotemporal dementia, and likewise. So, according to Alzheimer's Disease International report in 2019, 50 million people were affected with AD worldwide. And according to ARDSI, which is an organization in India, they reported that there were around 3.7 million Indians with dementia in 2010, which will become 7.6 million by 2030. So, India will have third highest caseload in world after China and US. AD causes symptoms of dementia such as memory loss, difficulty performing daily activities, and changes in judgment, reasoning, behavior, and emotions. And these symptoms of dementia are irreversible because it leads to permanent damage of the section of the brain. So it is a complex disorder, and the exact mechanism of action is not completely deciphered. So far, we only know that amyloid plaques and hyperphosphorylation of tau are the reasons behind Alzheimer's disease. So now let me tell you what is late onset Alzheimer's disease. So rapidly aging populations have increased the prevalence of dementia worldwide, and LOAD, late onset Alzheimer's disease, account for 80 to 90% of all the cases of dementia. And LOAD is currently regarded as the neurodegenerative disease, and epidemiological studies have demonstrated that diseases are associated with the incidence of LOAD. And while early onset AD is almost certainly genetically based, no specific gene mutations are associated with inheritance of the disease in LOAD. So the expression of the apolipoprotein E allele 4 is the one of the risk factors that are identified for LOAD so far, and to date animal models of Alzheimer's have focused on rare familial mutations, and due to a lack of frank neuropathology from models based on common disease genes. So that is where people are learning from it. So this is one of the neurodegenerative disorders. But let's go into the depth of one of them, which is Alzheimer's, and let's see what we have learned from the failures which have happened so far in the scientific fraternity. So you can see from year 2013 to 2018, these are all the medicines which were in the clinical trials for a very long time, and you can see that all of them were discarded. They were not brought to the market because of the lack of efficacy, toxicity, or they were worsening the cognition and increasing the mortality. So the reason, if we will see here, is that all of them are targeting something which is present in the brain, which is anti-amyloid beta monoclonal antibody based inhibitors, and likewise. So if you will see the clinical part of it, you will see the impact of you will see this research which says that if the resting mean is given long term, you will see that it causes the mortality in patients with Alzheimer's disease. So people are nowadays are not dying from Alzheimer's, they are dying from the treatment of it. So we can understand how difficult the nervous system is, and how difficult it is to work with the diseases which are from nervous system, right? So you can see another case report from a hospital which tells a fatal outcome after unintentional overdosing of histamine patches, which is a drug widely used for Alzheimer's. So now we need new drugs, and we need new drugs which go and bind at different places than brain, because we have had worked enough with brain and the receptors which are present in brain, and we see that in case of neurodegenerative disorders, we are not reaching anywhere. So let me tell you the latest trends in Alzheimer's disease, and you can put same in the other diseases also. So recently the studies came up, and they were mentioning the role of liver as one of the risk factors in cognitive decline and dementia. So now scientists all over the world are also working and looking at the role of liver in causing the dementia. And there are many studies which have published other than from the angle of the neurodegenerative disorders. Even the patients which are long-term patients of some liver illness, they are also experiencing reduced cognition. So this is how we know how brain and liver as an organ are so much attached and linked function-wise to each other. And there was a clinical trial wherein it was shown that a liver drug can also help to treat Alzheimer's. So now let's see why liver is important in neurodegenerative disorders. So let's talk about the liver-brain connection. Liver is important for brain because it supplies nutrients to the brain. It removes toxic substance from the body so that brain remains healthy. It is responsible for low-grade cerebral edema in case it is unhealthy. It maintains blood-brain integrity. It removes ammonia, and it is also the site of maturation of cholesterol in the body. And everybody knows the cholesterol hypothesis in Alzheimer's disease and how important it is. So liver and brain are therefore connected. So now there are many studies coming up recently from India and abroad wherein they are talking about the food and its impact on brain. So the things which I'm presenting in front of you right now, they are both the junk as well as the ones which come from natural sources. So let's see the impact of the food on the brain. So what you eat could be a factor in whether you contract Alzheimer's disease. Let's see. So there are many studies published which have shown the role of the dietary fructose in obesity and cognitive decline, and the food which is on the shelf these days, especially the ones which is sweet in nature and which has oil in it. So these artificial sweeteners are high, so they have high fructose and sucrose in them, and the high fructose and sucrose are not good for our hypothalamus, which is again the part of the brain which is responsible for keeping our memory intact. And also, several studies have come up which are showing that how the unhealthy oil and too much of sugar, obviously which comes from fructose and sucrose, that type is very detrimental for our blood-brain barrier, and hence can be contributing in the ill health of the brain. So this is how, since 95% of the dementia cases worldwide are sporadic Alzheimer's disease, which means you do not need to have a genetic history in order to have Alzheimer's. Anybody can have Alzheimer's later on in their life. So all these studies, they signify that if you will take care of your liver, probably there will be a less chance that you'll contract, dementia later on in our lives. So we really need to limit the bad food we eat so that we can keep our liver healthy. And if our liver is healthy, then definitely our, brain is healthy. So now modern research has revealed that dietary consumption of flavonoids and flavonoid-rich food, they significantly improve the cognitive abilities, and they inhibit and delay the aging process and related neurodegenerative disorders, which are also the part of the Alzheimer's disease. So if we will eat something wherein we have a lot of flavonoids, so we can take care of our body in totality and definitely the brain. So far the mechanism of the flavonoid has not been shown to be mediated through the inhibition of cholinesterases, including ACHE and BCHE, but they are implicated in cognitive and neuroprotective function. So flavonoids, they also hamper the progression of pathological symptoms of neurodegenerative diseases by inhibiting the neuronal apoptosis. So there are many good reasons why one should have flavonoids in their diet. And yes, of course, one should see a doctor before taking flavonoids in their diets. But one can always take those type of fruits and vegetables which are very rich in flavonoids, because that will definitely help, because flavonoids can reduce and can keep the brain healthy. So the next question is, where can I study neuroscience? So there are many places in India like University of Madras, Shivaji University, Ahmed University, Noida, University of Calcutta, NBRC Gurgaon, Rajiv Gandhi Paramedical Institute, New Delhi, University of Madras. And this is not all. There are many wherein you can go and study neuroscience in India. And yes, about the jobs in neuroscience. What do neuroscientists do? So neuroscientists, they conduct experiments on the structure, function, and development of the nervous system. They can work as research scientists for private research institutions or government agencies and hospitals. And they also develop new treatment procedures for neurological disorders. So what can I do to become a neuroscientist should be your next question. So step number one is you take science stream with physics, chemistry, and biology in your 10 plus 2 level. Then you can take up BSc or BTech in neuroscience from the universities, and then you can take up MSc in neuroscience as step number 3, and then you can do PhD in it, and then you can really go in the field of neuroscience. So now let's talk about the supporting groups and patients and caregivers. There are large, connected group of people, those who are joined with one or the other type of dementia and related disorder society, and they generate funds and then they invest in those NGOs who take care of the patients which come from dementia or other background. So, for example, in India, we have ARDSI, and Indian support groups have developed pool, and they support both the patients and the caregivers. So Alzheimer's and Related Disorder Society of India, it's in Hyderabad also, and it has a Calcutta chapter also. It has a Delhi chapter also, and then we have Echoing Healthy Aging, which is another organization based in Mumbai, and we have Nightingale's Medical Trust, who are based in Bangalore, and then we have another Sambedna Senior Care, which is also based in Bangalore. So if you want to go join them, you can. It is a good and noble cause. You can sometimes go meet the patients to understand more about the pathology, and likewise. So now time is running fast and we have to run with it, and we need to come up with new solutions to provide to provide a large section of population who is suffering from Alzheimer's. So now let me tell you the Nobel Prize winners in neuroscience so far. So this is the list of people who have been who have been getting the Nobel Prizes in neurosciences during all these years. So list starts from year 1906. So forth. So I hope this presentation was able to blow your mind. I'll seek your comments on the video. If there is something you want me to explore more in my coming videos, please let me know in the comment section. Thank you for watching.